we are going to listen Florian. We all know him from the first day where he introduced mass spectrometry. And today he is going to talk us MaxQuant output tables.
Okay, thanks for the kind introduction. Good morning, everyone. How many of you did go out last night? Nobody? Okay. So I guess you're very excited to hear now a beautiful talk about tables. And I can promise you we'll go through all of those tables one by one, by one, by one, by one. So so the first task is to find those output tables, and then hopefully during the talk I can get you a bit excited about the content and the wealth of those output tables. So one easy way to find them is to go back to the MaxQuant viewer that Jürgen introduced yesterday. And then you can notice this tab over there. That's the tables view. And there you can already get a first look at the tables. You can scroll through them. And it's actually quite convenient to get a first impression about your experiment. And then another way to find the tables is actually in the combined TXT folder. And there you find all the tables that were generated for your specific experiment. They're all in TXT format. And you can open them in Perseus or in Excel or whatever your favorite software for text files is. And then I just want to highlight this very last entry over there, which is the tables.pdf. And I highly encourage you to have a look at this PDF table if you're not familiar with the table, or if you find an entry that is not clear. So if you open it, you will find all the tables in the PDF format. And for every of the tables, there is a short summary. And then all the table entries, they're listed in another table. And you get a short description for what all of those columns, what they mean. So it's rather easy to get lost in all of those tables, but it's always good to have this fallback position. So whenever you get lost in the tables, go back to the PDF file and just get a first hint on what this column might tell you. So then to get started, then we'll just go one step back and actually have a look on how our experiment, the LCMS experiment, how does it look like? So as you now learned throughout the first 3 days, a typical proteomics LCMS experiment starts with the LC. Then we electrospray our peptides into the mass spectrometer, where we then acquire the data in a specific manner. So typically we prefer the data-dependent acquisition method, where we acquire full scans, which we use then to pick precursors for our MS-MS scans. And then those MS-MS scans, they give us a hint about the identification of the peptide. So what then we do with MaxQuant and Andromeda, we interpret those spectra, and we come up with a peptide sequence, which is then inferred to the protein sequence. And so we have different levels of our experiment. So the first level is the metadata. So this is very general. It gives you a rough idea about what you did, like all the general parameters. And then more on the mass spectrometry side, there is the level of scans. So this is actually what the mass spectrometer acquires. So it's MS scans and MS-MS scans. And then somewhat in the middle is already the first interpretation of MaxQuant. So this is this feature finder, a feature meaning this three-dimensional isotope pattern over time. So this is then the level of features. And once we have this set, we can actually identify those features by the MS-MS scans that are allocated to the features. So in total, we have 4 different levels of the experiment, and they are all reflected in the tables. So the tables are structured in a way that they reflect those levels of our experiment. So it's always good to think in this pattern when you look through the tables. And this is also the very same levels that you find in the tabs of the viewer, for instance. So you have the scans level with the identifications, the features, and the metadata. And then for all of those levels, there is certain sub-levels. So for instance, for the scans, we have the MS scans table, we have the MS-MS scans table, and if you did run an MS3 experiment, you will also find the MS3 scans table. So we can then go through all of those those tabs or levels. So I just mentioned the scans level. The next one is the identifications level, which is probably one of the most interesting levels for all of you. It contains the protein groups table, it contains the PTM sites table, but also it goes down to the peptides level, where we then have the peptides table, one table for modification-specific peptides, one table that contains evidences, and I will come back to this later and explain what evidences are. And finally, we have the table that contains all the identified MS-MS scans. Then one level above, it's this table that contains all the features. That's usually easy to find because it's always the biggest table. And also, if you click matching between runs, you can also get tables for matched features. And above everything, there is a summary table, one table that contains the parameters, and one table that is that gives you some information about the MOZ range and on what changed in between different MOZ ranges of the experiment. So we get started with that table that you're probably most familiar with, and that's in the identifications level, it's a protein groups table. So this is the perfect table when you're now like this. We're like, no, I don't want so many tables. I just want to get my protein identification and also my protein quantification. So this is the place to go for you if you now had exactly this in mind. So the protein groups table, it contains all the protein groups that were identified in your experiment. And throughout all the runs and throughout all the parameter groups, so everything is combined in this table. So there you find the protein-centric information. You find very helpful information about the physicochemical properties of your protein, such as the molecular weight, for instance. You also find a column that tells you how many counts or how many peptides you identified for this specific protein. You also get to know how you identified this protein. So was it via MS-MS or was it via matching between runs? You have a table that tells you the sequence coverage. So this can be very interesting if you look for very specific isoforms or something. So it's always helpful to check the sequence coverage of your experiment. And you get a summed number for the identified peptides. And finally, you also get the score information that come out of Andromeda. And even more important, you get all the quantitative information for the proteins. Meaning you get the summed extracted ion chromatogram intensity, you get the LFQ normalized intensity, or you can, depending on how you choose to perform your MaxQuant analysis, you can also get the IBEC intensities. Or if you did run a TMT experiment, you get the ratios and also the corrected ratios. And so for a label-free experiment, this is how this PDF file is going to look like. So it's going to list all the LFQ intensities for a given protein and for all the given experiments. So you get the intensity for experiment 1, for experiment 2, 3, 4, 5, and 6. On the other hand, if you run an experiment where you labeled the precursors or the peptides on the MS1 level, for instance in SILAC, instead of getting the LFQ intensities, you now end up with ratios. So this can be heavy to light, this can be heavy to medium, and quite conveniently, MaxQuant also outputs a column for the variability in this ratio, and also how many peptides did go into this count for the ratio. And also, if you check the requantify option, you also get an idea how many peptides were requantified and went into this ratio. And then if you run a TMT experiment, so you get even more columns, but so they refer to the raw intensities of your reporter ion channels. Then you also get, so the first one is the corrected by the isotope distribution of the label. So you get all the reporter channels for this non-corrected, for the corrected, sorry, and also for the non-corrected. So if you somehow find out that you put the wrong distribution in the correction parameters, you could actually do it here on your own, just by taking the non-corrected values. And again, also always very, very helpful, how many peptides did go into this ratio. So ideally, we have a lot of peptides, and then all of them make up a very precise and accurate ratio for the proteins. And the same is true for post-translation modifications. So there we have a very similar table. That's the PTM sites.txt. And that table contains all the proteins that were identified with a modification on one of the sites. So again, we have the information about the modified protein site, which means we get the localization probability and also the number of total modification sites. And as usual, we get the quantitative information also for the modified proteins, meaning we get the intensity. So this could be anything from label-free quantification to TMT. And we can also calculate the occupancy of this modification site. So if you're interested in this, I can refer to the talks later from Kirti. She will explain this in much more detail and go through everything that's relevant for PTMs this afternoon. So the conclusion from this tab, from the identifications tab, is that this is most likely going to be the two tables that you most of the time put into Perseus, and then you can do all of the bioinformatics post-processing that you now learned through the last 4 days. I mean, you can calculate differences between your samples, you can calculate the circadian rhythms of your proteins. So this is all contained in those two tables. So now we will leave the protein level and go one step down, which is the peptide level. So very similar structure, so that we also have all the information about the identified peptides. We have all the identifications of the modified peptides. We have those features that were identified as one peptide, and all of those features, they're the evidences. And then we have the MS scans underneath this level. So if you think about it, so we have proteins, and all of those proteins, they come from peptides, and then every peptide needs to have an evidence. An evidence could be a single MS event, it could be a match feature, but it can be several evidences for one peptide. And similarly, you can have several MS events that were identified for one feature. So it's like a cascade. So we can have a lot of MS events, and then they turn into evidences, and those evidences are then turned into peptide identifications. Peptide identifications infer protein identifications. So coming to the peptides TXT table, this gives you an idea how good or how bad your peptide identification and quantification is. So for all the peptides that were found, you obviously find the sequence in this table, but you also get all the score information about every single peptide. So you could, for instance, plot the score distribution of all your peptide identifications and then compare it between different days or between different experiments. And also it gives you all the physicochemical properties of the peptides, meaning the mass, how many amino acids are in the sequence, what is the first amino acid, what is the last amino acid, and also quite interesting, you get the cleavage windows of your protease. So this can be a very, very interesting quality control for your experiment or for your digest. And then additionally, we have protein-specific information in this table. And so this is a very important feature of all the tables in MaxQuant. They are all linked together. So if you open one table, you can always link it to another table by identifiers. So for instance, here you could link your peptide information to the protein if you link it through the protein-specific information. So you get all the links in the tables. So in this case, it would be an identifier for the protein, but you also get a column that tells you the position of the first and last amino acid within the total protein sequence. And finally, we can have the quantitative information. So this can be very interesting, for instance, for TMT experiments. So instead of now looking at the quantification at the protein level, you can also look at the quantification on the single peptide level. And this can contain much more information than the sum protein ratios. So one use case for this peptides TXT table is, for instance, if you found a vial in your freezer and it contains a label that you can hardly read, but it might say something about protease, So you just put it on your cell line of interest, and you want to figure out, okay, is this still working, or has it just been lying around for too long? And in this case, the peptides.txt table can help you. What you can do, you can plot the distribution of your peptides. So is this something what you expect for triplet peptides? So there should be they should have like 10 to 15 amino acids on average. So in this case, it looked quite good. And you can also look at the miscleavage rate. So how efficient was my digest? Like, did I cleave all the peptides, or do I have a lot of miscleavages in between? So I mean, the protease didn't work, or was probably too old, or not concentrated enough. So this is very easy to plot from this table. You just load it into Perseus, and it's probably 2 clicks, and you end up already with a graph like this. And a bit more advanced, you can also calculate the cleavage windows of this protease. So in this case, it looks like it was a triplet digest, but this can apply for every other protease that you want to try. So for instance, if you read the latest paper from Albert Heck, and you think, I want to try a different protease, so this is a good strategy to check the digestion efficiency and also the specificity. And you can find it all in the peptides table. Another use case for this peptides table is when you listen to Tammy's talk on Monday, I think, where she introduced peptide fractionation. So now you get very excited about fractionation. You will definitely want to try it when you get back to the lab. And you run this first experiment, and then it can be hard to assess the quality of the fractionation. So meaning, how many peptides did you get in a single fraction, or did all the peptides spread over 2, 3 fractions, meaning that your fractionation was not very sharp and efficient. Again, that's very easy to get from the peptides table. So you can, for instance, cluster them in a histogram or in a plot like this, and then you see you can color code the intensity, meaning that red means that the peptide there was abundant, and in green it was not abundant or just very low abundant. And then you can cluster the different fractions, and then you see that most of the peptides, they ended up in 1 or 2 fractions, which is actually a reasonable result, but maybe you can improve it on your own. So that's, again, easy and nice quality check for fractionation experiments. And then coming back to this level of evidences, so as I mentioned before, we step down from the protein level to the peptide level, and then all the peptides identifications, they come from evidences. So an evidence can be an MS-MS event that was successfully identified, or it could be a successfully matched feature. And this comes back from the feature detection in the first part of the experiment. So the evidence is already quite down all the way to the mass spec. So this is, now we're getting closer more and more to tables and information that are really linked to the mass spectrometry performance. So what you can read from this file, and you get it for all the single raw files. So that's probably a difference to the other tables. So the other tables, they were all based on the experiments as you define them in the layout in MaxQuant. But now in the evidence table, you get all the information resolved by the raw file. So sometimes this is not exactly the same. So you have to think about how to use and interchange parameter or the information that you get from those 2 files. So you get resolved by the raw file, again, all the score information. You get all the information about the feature, meaning the mass to charge ratio, the retention time, the calibrated retention time. You get a column that tells you how many MS-MS events were acquired for this specific feature. And if you want to go back to your raw file, you can also look at the MS-MS scan number column. So this number that you find in this column is exactly the same number that the scan is given in your raw file. So if you want to open your raw file in, for instance, Excalibur, you can navigate to this scan via the MS-MS scan number. And you get information about the recalibration. So was there a mass recalibration involved? Was the retention time recalibrated? And also if it was matched, it will give you the matching time difference between the feature in this specific raw file and the feature where it was matched from. And it will also give you the master charge difference. So you can already judge the quality of your matches just by looking at the time difference and also the master charge difference. So if both of them are very high, then it's unlikely to be a good match. But if both of them are very, very tight, then you likely have a very good match and you want to go on with it. And as mentioned before, so this table now links the raw files to all the other output tables. So this can be very, very helpful. So just imagine you were measuring your runs over the weekend, you enjoyed the free time at the beach, and then you come back Monday morning and you find an email like this. So this says, "Hello together, sorry, we have an error on our cooling units in all MS rooms." Perfect. So meaning that probably most of your runs from the weekend, they just went wrong because the air condition failed over the weekend. So if we were just acquiring the last files for your nature revision, this is probably not the email you want to get on a Monday morning. So then the question is, how can you figure out if your files were affected that you acquired during this time? And do I have to repeat all the experiments, or do I just want to look into the latest experiments? Because you only know that it happened during the weekend, but you don't know when. So it could be on Friday night or just Sunday night if you were lucky. And there it's a good advice to look into the evidence TXT. And everything that is related to temperature changes is usually reflected in changes in the mass accuracy of your experiment. So a good idea in this case is to simply plot the uncalibrated mass error versus the retention time. So this was a 120-minute run, and then Perseus has this nice feature of just by one click you can calculate the densities. So this is very, very helpful. So this blue and whitish region over there, that's where most of your data points are. And you can see that they're nicely centered. In this case, I think, what was it, like 8 or 5 ppm off, which is okay because this can all be recalibrated. So let's say this was your run on Friday night, and then you check another run from Saturday morning, for instance, and then it looks like this. So immediately you see that something went wrong in this experiment. So this could be because of the air condition or some other instrument failure. And it's just 2 clicks to figure this out and track down the problem in your runs. Another use case, especially when the temperature changes, also we often see sometimes changes in the retention time or in the stability of your chromatographic performance. So it's very easy to plot the evidences from file 1 versus the evidences from file 2 and just check if the retention time changed. So ideally it looks like this, so that most of the retention times are on one line and only a very few outliers, and you can. So this is probably the scenario that you want to have. And again, if something fails, you can immediately read it out from such simple plots within just a few minutes. And also, maybe you've been trying different columns, and so you can also plot some histograms and see what was the width of your chromatographic peaks. So for instance, here it was not that hard to figure out that column 1 was actually performing the best. So this gave very sharp peaks and with a medium retention time of about 30 seconds. And then columns 2 and 3, they were obviously not that good. So they generated very broad peaks, and some of them were more than 2 minutes broad. So this is probably not the column that you want to use for your further experiments. So you might think about changing your column, and you can also track this during your experiment. So you have a 2 weeks run, so at some point you might want to change your column, but it's not exactly clear when to do this. So from time to time you could just run some of the files in MaxQuant and check how does my chromatographic performance change, and do I have to do anything about it. Okay, and another thing is this dynamic exclusion. And I Googled for it last night because I wanted to have a nice figure or some picture that explains it. But if you actually Google for a dynamic exclusion, you find those 3 cute dogs. So, not sure how much this is going to tell you about dynamic exclusion. But it's a parameter that we set in our mass spec method to avoid resequencing of MS features. So meaning that you're happy with just having one MS event per feature, because this gives you the maximum depth of your proteome. So you want to avoid situations where you sequence the very same feature over and over again. And therefore, we set this parameter called dynamic exclusion. And this puts every feature that was sequenced once on a list, and once it's on this list, it's excluded for the next X seconds. And then you can define the time for which this feature is going to be excluded from sequencing. So, and this should be related to the chromatographic width of your system. So you can use the table from before to get an idea of how broad are my peaks, and then ideally you want to set the dynamic exclusion in this range. So it should not be more than your chromatographic peak width, but it should also not be significantly less than your chromatographic peak width. So you can get the first idea from the evidence table and see what width should be good for your experiment. And then you can also plot exactly this number. So the count of MS events per identified feature in your next experiment, and then ideally it looks like this. So most of the features, they were acquired with one MS event, and only a very low percentage of features was acquired with two MS events per feature, and then none of them had three MS events per feature. So then the question for you is, how can I have identified features with 0 MS events? So what does that tell you? Yeah, exactly. So those are the matched features. So you can also identify them by looking at this column and then draw your conclusions from there. Okay, now we are leaving the identifications level and we're moving even closer to the mass spec, and we go to the scans level. So there we have the MS scans table, the MS scans table, and the MS3 scans table. And it's actually linked to the MS.txt table. So don't get confused. If you find both of them, they sound very similar. But the difference is that the MS.txt table contains all the identified MS scans, and the MS scans table contains all MS scans. So regardless if they're identified or not. So ideally, this is shorter and easier to open than the full one. So as I mentioned, the MS.txt contains the identified MS events. So there you have the fragmentation type that you set in your parameter groups. You get the number of filtered peaks. We'll come back to that later. You get the number of matched fragment ions in this specific spectrum. You get all the annotated peaks and also the intensity fraction, and also the mass deviation for every single fragment in this experiment. And then one level above is the MS scans table, where you get more information about the acquisition. So this also includes the ion injection time and the AGC fill in case of orbitrap mass spectrometers. Meaning the ion injection time is the time that you use to inject ions from this precursor to the C-trap and the HCD cell. And the HEC fill is a parameter that's output by the thermal raw file, and it gives you an idea or a fraction of how much of your HEC target you reached. So if this is 1, that means that the HEC target that you set in your experiment was most likely fulfilled. If it's 0.1, meaning that you were working only with 10% of the number of ions that you initially wanted. And then I will now spend a bit time on those parameters or columns below. It's precursor apex fraction, precursor ion fraction, and the precursor intensity fraction. So the precursor apex fraction gives you an idea of how close the MS event was to the apex of the chromatographic peak. So ideally, we want all our MS experiments to happen at the apex of the chromatographic peak, because that's where we expect most of the ions. So ideally, we always trigger the MS event on top of this peak. And then the precursor apex fraction gives you an idea of how close you were to this ideal position. So if it's 1, it means you exactly triggered the MS event on the apex. If it's 0.8, you are still good, but not ideal. And then if it's 0.1, you probably should have waited a bit longer to acquire this MS experiment. And a very, very similar column is the base peak fraction. So this gives you an idea about the dynamic range that you cover with your top N experiment. So meaning this is the fraction of the peak that was triggered, or of your precursor peak that was triggered for MS in relation to the most abundant peak in this specific survey spectrum. So in this case, if we would have triggered this peak at MRZ 652, we would have gotten a base peak fraction of 1.0. And this one would have been 0.5. And then one of the lower abundant guys over there would be less than 10%. So this gives you an idea of how much of your peptide space do you actually cover with your experiment. So if you always end up with numbers close to 1, you probably should try to increase the speed of your MS experiments. On the other hand, if you have a lot of low abundant peaks and you don't identify them, you might think about slowing down your experiment and leaving more time for the mass spec to acquire MS data. And then one thing that came also up during this week is this precursor ion fraction. And this is the fraction of your target ion within the isolation window. So unfortunately, we cannot set the quadrupole to isolate just one single peak, but we always have to give it a window. So typically, this can be 1 Thomson or a 0.8 or something like this, meaning that there is not only your target ion within this window, but you also isolate other peaks that just by accident happen to be in this window at the exact same time. And then this precursor ion fraction gives you the relative ratio of the target ions versus all ions. And this is very important for your TMT experiments, because when you have a situation like this where you have a lot of co-isolated peaks, this means, in particular on MS2 level, you run into this problem of ratio compression, where the TMT ratio that you read out from this event is not only coming from your target ion, but also from all the other ions that bleed into this isolation window. So you could run one TMT experiment or a regular experiment in this way, and then look at the distribution of your PIF values. And you can actually use this to then later on define this filter in the MaxQuant experiment for your next runs. So for instance, if you figure out that all of your ratios that you get from precursors with a PIF value below 0.5 are not very useful, you could set this filter to 0.5 and you would already get rid of all of them. So this gets you around this manual filtering step afterwards. You can already filter it here before you even start the analysis. Another obvious question you could ask is, how good are my MS spectra? And then there's this interesting column that's called filtered peaks. It's more of an empirical observation that if you have identified MS spectra, they tend to have more filtered peaks than unidentified spectra. So I cannot really explain why this is like this, but it's more of an empirical observation that we did throughout our experiments. So the number of filtered peaks is actually the number that goes into the Andromeda search in this iterative procedure. And if you have more filtered peaks, that actually indicates that there were more peaks and a broader MROZ range covered in this MS scan. Well, just an interesting column that if you're running QC experiments, it can be very helpful to monitor this over the period of your experiments. And then also, so the quadrupoles, they tend to get a bit dirty after some experiments, meaning that you transmit less ions than you did initially. So if the field is distorted, then the ions are not as efficiently transported through the quadrupole, meaning we detect less and less ions, even though we need every single ion to get a nice MS identification. And again, if you're running QC experiments, this is something you can, for instance, plot in Perseus. And so one idea is to plot the precursor ion intensity versus the total ion current in the MS2 event. Ideally, this would be 1, because then that would mean that all the ions that were in the precursor were successfully transferred into the MS-MS event. And then you can monitor this. Not sure if the absolute number is telling a lot, because there's some other factors going in there. But if you monitor this over several weeks or months, and you suddenly see that this cloud here is going down, then this could indicate that your quadrupole is getting dirty, and you might think about cleaning it. Similarly, you could look at the ratio of the base peak, meaning the most abundant peak in your MS-MS scan, versus the total ion current in the MS-MS scan. And this gives you an idea about the fragmentation efficiency of your MS-MS experiment. So meaning if all your ion current is concentrated in the base peak, so you can imagine a spectrum that has just one single peak, meaning you did not fragment it at all. On the other hand, if the base peak is very low, then you might have over-fragmented it. So this is another parameter. So if this suddenly changes, then you know there is something wrong maybe with your HCD cell, because you no longer fragment, or you lost the gas, the fragmentation gas in the cell, or something like this. Okay, so coming from the MS-MS scans to the MS scans, which are our survey scans where we pick the precursors from. So this is again coming closer to the raw file, so it gives you all the information about all the MS scans throughout your experiment. It gives you the scan number, the retention time, the ion injection time, and also how many ions were in this scan. And you can, for instance, use it to plot the cycle time of your experiment. Or you can plot the number of MS events that you acquired throughout the experiment. So one question that I often get asked is, we always talk about those top n methods, but how many n's do we actually need, and what is a good number of n for my experiment? So one guideline for this is that you want to keep the cycle time low enough to cover the chromatographic peak, so that you have at least 6 or ideally 10 data points or MS-MS scans over a chromatographic peak. And this then gives you a hint on how many MS-MS events you can squeeze in between 2 MS events. So we prefer cycle times of around 1 to maximum 2 seconds, and we just squeeze in as many MS-MS events as possible. So you can read out the cycle time. So here in this case it was even below 1 second, and it's in 2 different experiments, so this looked all very reproducible. And we at the beginning of the peak, you see there is not many precursors, so we only perform a few number of MS-MS events per cycle. But then later, when all the peptides kick in, we end up with a top 15 method. So it's also interesting if you set this n parameter to 100, for instance. Actually, through the threshold filtering, you're never going to reach the 100. So it's a nice experiment just to set it to 100 and see what is actually the maximum number of precursors that you could find in your MS scan. Okay, so leaving the scans level and also leaving the identifications level, we move to the features. So the features, those are all the isotope patterns that were detected with this 3-dimensional algorithm. So meaning you have at least 2 isotope peaks, and you have it in at least 2 consecutive scans. And this is then what MaxQuant bases the quantification on. And also generates the accurate mass. So for all the features, we get the information about the mass to charge. We get the charge, we get the intensity, we get the retention time, and you can also plot things like the peak length, and you can also plot the shape of your chromatographic peak. So it will give you a start and an end time of the chromatographic elution. And if you then know the apex, you can see if your peaks are distorted or not. And you can also plot this well-known histogram where you plot all the features from your experiment. So this is the gray distribution in the back. So it's plotted as a histogram of the logarithmic intensity. And typically, this looks like a log normal distribution. So we have a lot of features, and they cover a dynamic range of 4 orders of magnitude. And then you can color code those features that were actually targeted for MS-MS. So in this case, those would be the red features, and you can also color those that were successfully identified. And this then tells you a lot about the dynamic range of your experiment, and also a bit about the problems of data-dependent acquisition, meaning that we have this slight bias toward more abundant features because they are easy to identify, so we're actually quite happy to sequence them. And then also for the identification, so it's easier to identify those high abundant features, and then for the low abundant features, we lose a bit in our identification rate. So you can also use some of these plots to optimize your method and see if you want to cover more of this gray population, meaning you need more MS-MS events, or if you're not happy with the identification rate, meaning you probably want to go for less MS-MS experiments and spend more time to acquire ions and increase the chance of identifying a peptide. Okay, last but not least, we arrived at the other part of the spectrum, which is the metadata. And there we have a table for a summary table that summarizes your experiment. We have one table where you can look back into the parameters that you set. So those are all the parameters that you defined in the MaxQuant search. It's always good to look back into this table if you were not sure about how you set parameter X or parameter Y. So you can always look it up in the parameters table. And then there's also one table that is probably not frequently used, but this summarizes information from the other tables, but now it gives output as a function of the MROZ range. So I think there is some use cases where this could be useful, but it's probably nothing that you would want to use in your daily life. So again, coming back to this quality control, because I think if we run those experiments, it's very important that we make sure that all of our experiments are with the best quality that we can do. So you want to make sure that the mass spec was performing well, that the LC was performing well. And so now if you are done with your MaxQuant analysis, probably the first step will always be to look into the summary TXT, because there you have all the information combined, and it gives you a very quick idea of how good were my runs, was there something wrong, and which runs went wrong. So you get all the information at one glance, and you can have a look at it. So meaning for all the experiments and for all the raw files, you get the number of identified peptides, the number of detected isotope patterns, the identification rate of the MS-MS events, you get the resequencing rate, and you also get the mass deviation. And so if something is very far off in one of the experiments, you might want to go back and look into the raw file and see what actually happened there. Because you probably do not want to include such a file into your quantification or use it for your biological conclusions. And this is just an example from our lab. So every week for all of our instruments, we run QC runs. So this is a triplet digest from HeLa cell line. And we plot them over the entire year, and I think now we have a record of, a few years. And it's relatively easy to plot all of those parameters. And those parameters, they have a problem that there's no clear cutoff, but it's very helpful to plot them over time. And so if you run your QC runs in a regular manner, you can plot all of those parameters in columns over time, and this will give you a very good hint about the performance of your mass spec and when it's time to clean it or not to clean. So we used to do this in a regular Excel sheet, but I think now there's also several papers from other groups that plot several of the MaxQuant tables in R or other scripts, and you can easily import them, and then you get an output about the quality control parameters of the mass spec and also the LC. Okay, so coming to the end of the talk, this is just the overview again. So again, I hope you get familiar with this concept of the different layers. So where we have the scans layer, which is probably the closest to the mass spec instrument. Then we have the identifications level, where we get the protein identifications and also the peptide identifications and all the quantitative information that you've been looking for. Then we have the level of the features. So you get the number of features that were detected on the MS level. You get the number of features that were identified and how they distribute over master charge and over the retention time. And then on top of all of this, there is this metadata table that gives you a very quick view about the performance of your experiment and the parameters that you use for the MaxQuant search. So hopefully you get very excited about all of those tables, and I can highly encourage you to look into them, and it's really great. It's a lot of information and they're all there, and I think you only acknowledge them if you actually miss some of those tables, because then you lose a lot of the information. So proteomics experiment is much more than just looking into protein groups and looking at different information. So I can encourage you to look in those tables and find your way through it, and always remember the tables.pdf file to, whenever you get lost, to come back and get a short description of whatever the column you're looking for. However, if you're still not convinced, you can also use this function in MaxQuant and just turn some of the tables off. So it can be very helpful for large data sets where you can just uncheck some of the tables where you're absolutely not interested in, but hopefully you're never going to use these functions. Thank you.